A metabolite is in fact a small molecule that is produced by living organisms. Plants are kings of producing small molecules. They produce thousands and thousands of them. Humans perceive them as taste, flavors, aroma compounds, pigments, and different coatings of plants. Metabolites are in fact the notion of the plant. This is what makes a rose smell like a rose and a tomato taste like a tomato. The word metabolism comes from the word to transform in Greek. These small molecules are produced in a way like the subway, the trains that are moving a molecule from one station to another, taking a substrate and making a product in a way that will, in the end, we will have a product such as a pigment or an aroma compound. Metabolites will impact our daily life since they are really produced by any plant species, and we will consume them or interact with them in some sort or some way every day. There are many drugs, vitamins, and other commodities such as pigments and aroma compounds which originate from plants.
Metabolites are not only important in plant research, but they're also very useful as a biomarkers for different human disease. We specifically collect blood samples from children with depression and anxiety, and we try to predict using metabolites or metabolic profiling in the blood of these kids the chance that they will develop severe side effects due to the treatment.
One of the group of metabolites that we have been studying are pigments called betalains. For us, these molecules are really strong antioxidants. They are known as supplements that we can use to improve our health. One of the intermediates of this pathway that leads to the production of the pigments is L-DOPA. L-DOPA is a very important drug for Parkinson, and in fact now we have in hand the way to produce L-DOPA to very high levels and purity in other systems by engineering. We have been studying potato, and this is common knowledge that potato, when they get green, they form these so-called solanum alkaloids which are toxic to humans. And nowadays, using metabolomics tools, we can make the potato new varieties of potato which will not make this toxic molecule anymore, and the potato could be consumed without any problems.
You've all heard of genomics, the genes that are all collected together in one organism. You've all heard maybe of transcriptomics, and that's the expression of those genes in an organism. The third leg is proteomics, which is a translation of those transcripts to proteins. And the very important leg, which was missing until now, is the metabolomic leg. And that leg actually is what makes the whole table solid and standing, so that someone can go from the genome all the way down to what actually the gene is doing at its most basic product basis. And this is revolution.
Before the era of mass spectrometry, people could only work and study only a few molecules. What metabolomics allows us is to really look at hundreds and thousands of molecules in one go. Mass spectrometry is really the best tool we have today to detect and identify these molecules, reconstruct it back, and then understand what is the identity of these chemicals. In our lab, we in fact developed a very nice system which allows us to track and monitor thousands of these molecules using multiple instruments that are based on mass spectrometry.
The potential use for metabolomic profiles is enormous. All the scientists today who look into genomics or into transcriptomics are the same sort of people who want to know what the metabolites are.
We, for example, collaborate with scientists in the field of nutrition and the field of personalized medicine. Learning how to engineer these metabolites is in fact the future of human health.